We are now ready to start talking about cells. The study of cells is called cytology. So the prefix cyto stands for cell and ology stands for the study of. Now, again, coming back to our theme of unity and diversity, right? We know life is different, but we know that different organisms share certain points of similarity. And so on this slide, we identify the basic features of cells that every single cell has, regardless of what organism it belongs to, if it's prokaryotic, eukaryotic, unicellular organism, multicellular organism, doesn't matter. All of these four components are something that all cells have. So first of all, they have a plasma membrane, right? They have to have some sort of boundary that separates the cell from its environment. Number two, we've got the cytosol. So the cytosol is a semi-fluid substance that's on the inside of all cells. It's a semi-fluid substance simply because, yes, it's an aqueous or a water-based solution, but there's so much stuff dissolved in it that it makes that fluid really quite thick. Number three, we've got to have chromosomes, right? We know that all cells, with a few exceptions, have DNA, right? And they have to have that DNA. They have to carry those genes so that they know how to behave. An exception would be a red blood cell. A red blood cell doesn't have a nucleus. It's not going to have any DNA. This is why they don't last very long, and you have to continuously replenish the red blood cells in your blood using bone marrow. Okay, and then lastly, number four. There's no point in having genes in a cell if you can't express the information that those genes code. And quite a few genes, probably the majority of genes, actually code for the production of proteins. So you have to have the machinery to make proteins if you're going to make use of the genes that you got. And so the machinery that makes proteins in cells, these are called ribosomes. Ribosomes are not considered organelles. They're not bound by a membrane, and all cells have ribosomes, irregardless of whether they're prokaryotic or eukaryotic. Now, maybe you've thought about this before, but we understand that bacteria are really small unicellular organisms. Why is it that a human or a dog or a cat, for example, is a multicellular organism? Why can't we just be one big blobby cell? Why does that not work in nature? So let's talk about the size limits for a cell. How small can a cell be and how large can a cell be? On the terms of, on the small side, a cell can get really very small. Prokaryotic cells are teeny tiny, sometimes even half a micrometer. And they can be that small because they don't have a lot of material on the inside, right? They don't have any organelles, no nucleus, nothing like that. And so they can be smaller. A eukaryotic cell is going to be a little bit bigger. It has more components, more DNA to house. So pretty much a cell can be as small as it needs to be, so long as it can still carry all the materials that it needs to be able to survive. On the other end of the spectrum, how big can a cell get? Well, this really depends on the surface area to volume ratio on a cell, right? We talked about the membrane and how the membrane is this boundary between the inside of the cell versus the outside. But not just being a boundary, the membrane also regulates what passes into and out of the cell. And so a cell needs to have nutrients coming in and waste coming out. It needs enough nutrients so it can survive. The only way that the nutrients get in is going to be through the plasma membrane. So you have to have enough of that real estate, enough of that plasma membrane to get enough nutrients in to keep the cell alive. So this comes down to surface area to volume ratio. Let's go through a couple of examples, right? So we've got this teeny tiny little cell, right? Let's say it's a cube and it's 1 unit per side. If you were to calculate the total surface area, that would be a surface area of 6 units. If you were to calculate the volume, 1 times 1 times 1, right, length times width times height, you would have a surface area of only 1. So if you compare surface area to volume ratio, 6/1, you'd get a surface area to volume ratio of 6. That doesn't mean anything yet, but let's compare it, let's say, to a bigger cell. What if a human, for example, was a big blobby cell like this? And this cell is bigger, having 5 units per side. So when you calculate the total surface area, yes, it has more membrane, 150. And then if you calculate the volume, also the volume increased. It's a larger cell, you have 125. But if you compare how much surface area, how much plasma membrane does this bigger cell have in comparison to the volume of cell that it needs to feed on the inside, that surface area to volume ratio fell drastically. It's only 1.2 now, as opposed to the 6 for the smaller cell. This means that this smaller cell here has a lot more plasma membrane or cell membrane to get nutrients in to feed a smaller volume on the inside than this larger cell. Maybe this larger cell doesn't have enough of that plasma membrane to get enough nutrients in to feed that internal volume of the cell, and the cell is just not going to survive. It's going to starve. So this is one of the reasons why we as humans, we can't be one big blobby cell. We wouldn't get enough nutrients across our cell surface to be able to survive. So instead, we're divided up into trillions of cells to fill that same volume, right? So you have the big cell here. What if you take that same volume and now you just divide it into single cells that all sort of function together? In this case, if you were to take the total surface area of all those tiny cells in that volume, you'd have 750. The total volume is still the same as this larger cell. You just divide it up into smaller cells. And so when you now compare that surface area to volume ratio here, you're back up to 6. You're going to have plenty of that plasma membrane to get nutrients in, to remove waste, to keep the cell alive. Okay, the next couple of slides are going to give us probably a review and maybe a reintroduction to the different kinds of cells that we have, right? So we have two main categories of cells. We talked about this in chapter 1. We've got prokaryotic versus eukaryotic cells. And so what we're going to do is we're going to review them and their basic characteristics. So first of all, prokaryotic cells. We know that these are small. We know that they're very simple. We know that they don't have any membrane-bound organelles, so that means they're not going to have a nucleus. They still have DNA, right? They still have ribosomes. They still have a plasma membrane. They still have cytosol, all those things that the cells have. They just don't divide it up into internal compartments. Everything's just floating around on the inside of the membrane. The DNA, while it's not membrane bound in the nucleus, it is located in a region called the nucleus. So that's just sort of the area where the DNA hangs out. Oftentimes it might actually kind of be anchored to the plasma membrane. So you can see if this is our prokaryotic cell cartoon. In blue here you've got the chromosome, and this area where the chromosome is hanging out, that's called the nucleoid. Now we also introduce another word that starts with the letter C. So far we've talked about cytosol, right? Cytosol is going to be the semi-fluid substance on the inside of cells. The cytoplasm sounds very similar. The cytoplasm is actually the cytosol plus anything else that's dissolved within the not just dissolved on the inside of the cell, but also hanging out on the inside of the cell. So, for example, in this prokaryotic cell, literally everything on the inside of the membrane is part of the cytoplasm. So that would include the chromosome, that would include the cytosol, that would include the ribosomes. So if we were to draw that out, right, so for a prokaryotic cell, so if we have a prokaryotic cell, right, and let's say blue is my plasma membrane, the cytosol is going to be, the cytoplasm is going to be everything on the inside of that cell. So everything in red is going to be the cytoplasm. Okay, we'll come back to this in just a minute. Now, remember that the life on our planet can be divided up into 3 different domains, right? You've got your domain bacteria, domain archaea, and domain eukarya. Two out of those 3 domains are made out of these prokaryotic cells, and those include any organisms that belong to the domains bacteria and archaea. Now, let's go ahead and review eukaryotic cells. The 3rd domain of life, the domain eukarya, is made out of eukaryotic cells. We think that these evolved later. They're more complex in their internal structure. They do have membrane-bound organelles, so they're kind of like little organs on the inside of the cell. They've got a membrane. It's like a little sac that has a particular function. The nucleus is an example of an organelle. In the nucleus, its main responsibility is to hold the DNA for the cell inside of that sort of membrane-bound region. Eukaryotic cells also have cytoplasm, but it's defined a little bit differently. So let's go back to the drawing. Right, so that was our prokaryotic cell. For a eukaryotic cell, right, we have to draw two membranes, right? We've got your cell membrane on the outside, and then we also have the membrane that holds the DNA in the center, right? That's the nucleus. In a eukaryotic cell, the cytoplasm is defined as everything in between those two membranes. So everything in between the plasma membrane and the nuclear, what we call envelope, or the nuclear membrane. So it kind of looks like the donut, right? A donut. So a little bit different from what you have for the prokaryotic cell. Okay. We talked about how eukaryotic cells are generally larger than prokaryotic cells, and protists, fungi, animals, plants are all made out of eukaryotic cells, right? All of these organisms belong to the domain eukarya. All right. And then the last thing we probably need to cover before we dive into the details of cell structure, when we're talking about eukaryotic cells, right, we know that the eukaryotic cell is subcompartmentalized, right? It has all these little internal membranes that partition the cell into these different organelles. And the organelles being membrane bound, right? They have a membrane that surrounds them. These biological membranes are really no different than the cell membrane itself. And so we'll talk about membrane composition and function when we get to chapter 7. But it's essentially a double layer of phospholipids amongst other lipids and proteins that give that structure to the organelle membranes. Second half of this slide talks about comparison of different cellular structures that plant cells have, maybe the animal cells don't, and vice versa. So for plant cells typically have a cell wall. We're going to take a look at this. Cell walls are made out of cellulose. This should be familiar when we talked about structural polysaccharides. Back in chapter 5, we talked about cellulose and how it's a very rigid carbohydrate that gives a cell wall its internal structure. And the cell wall, again, helps to protect the plant, the plant cell, and give it some rigidity. Plant cells also have to be able to communicate with each other, right? When you have plant tissue, it's made out of multiple cells. Those cells need to talk to each other so that the plant can survive as one organism. And so plant cells are going to have these plasmodesmata. There are communication channels between adjacent cells that allows them to exchange small molecules and essentially to communicate with each other. Plant cells also sometimes have the ability to contain chloroplasts. Chloroplasts are the organelles that perform photosynthesis, so they create sugars for the plants. And then lastly, plant cells also have something called the central vacuole. Now, there are different kinds of vacuoles. Plants specifically have a central vacuole, which is a really large sort of holding tank on the inside of the cell. And so those are all components that animal cells lack. On the other hand, what do animal cells have that plant cells lack? So animal cells have lysosomes. This is like the stomach of the cell. Your stomach helps you digest food, has a really low pH. Well, a lysosome in an animal cell helps digest macromolecules and has a really low pH. Same thing. Animals, their cells also oftentimes will have centrosomes containing centrioles. We're going to come back to this guy a couple of times throughout this semester, but essentially a centrosome is an area in the cell that helps to build the cell's skeleton. It builds these things called microtubules, and sometimes the centrosomes are going to hold these structures that are called centrioles. Centrioles are also little microtubule structures inside of those centrosomes. And then lastly, animal cells are more frequent to have flagella. There are some plant cells that have flagella, but this is more frequently found in animal cells.